At the University of Georgia, we have a number of faculty that are involved all the way from cutting-edge assays for cell biology as well as engineering. And by being able to bring them all together into CMaT, we can address really focused interests and focused problems associated with cell manufacturing.
Our research is broadly in the field of metabolomics. Our lab measures small molecule metabolites from living systems, whether that be cells or model organisms or humans or plants or other material. And using that information, we figure out what's going on inside the cells or organism that we're studying.
So we use NMR to measure the metabolomics profile of stem cells, mesenchymal stem cells, and that metabolomics profile we believe can be used to predict its potency. So we can certainly identify the critical quality attributes from the metabolomics profile.
The NMR facility that I direct in the CCRC is fabulous. We have some of the best instrumentation found anywhere. We have instruments that are capable of some rather unique measurements. We have very high field strength NMR, medium strength NMR, and high throughput capabilities. And so basically we have state-of-the-art equipment for metabolomics, for NMR metabolomics. So we're working with cells that Rooster Bio has provided to our collaborators, Stice and Ross Markland. And then we work very closely with Evolved Analytics on data analysis of the very complex and heterogeneous data that our projects have been producing. And so that's a really critical partnership.
Overall, we work mostly in the area of what's called super resolution fluorescence microscopy. And these are sort of a host of techniques that are designed to give you resolution beyond the diffraction limit with a conventional fluorescence microscope. So that means we're trying to achieve resolutions between 150 nanometers down to around 20 nanometers. Whereas a typical microscope will give you about a 250 nanometer resolution. An area I'd wanted to get into that sort of is complementary to some of the research we do is called is some of these new techniques for phase microscopy, or also it's called computational imaging. And this is a whole series of sort of very new cool techniques where you can use relatively low cost light sources to illuminate samples in a variety of ways. And from those multiple images, you can then reconstruct very sort of high resolution phase maps of the sample. I mean, the goal of our part of CMaT is to develop sort of a microscope that can do sort of real-time, inline cell assessment, so that as cells are being manufactured, they can be imaged with our microscope, and then so machine learning algorithms will run to decide, what the quality of these cells is. And that sort of complements nicely some of the other techniques that other groups are developing as part of CMaT, which are sort of more invasive and give you maybe more detailed information, but can't be used in line, take more time. You can't then take the cell that you imaged and then go ahead and use it because it's a destructive measurement. So we're really sort of providing a complementary technology.
What we all want to do is have our technology be used by people, particularly in the area of cell therapy, where again, the cost of therapies are huge, the uniformity is a problem. All of those are real problems that takes an entrepreneurial spirit to solve. And at the University of Georgia, all the activities that are associated with our new innovation district will really guide and nurture those capabilities.
Over the past 5 years, what we've tried to do is increase the number of people who are involved in helping this process move forward, bring in more people who've got actual experience of startups or of commercializing technical inventions. We've expanded significantly the number of programs that we operate here at UGA. So we've added iCore to allow people to do customer discovery, to actually see whether there is a real need for what they do. But on top of that, we've added many different programs to just increase the level of knowledge that people have about not only what the outside world looks like, but also what resources are available to them within the university. And finally, there's been a significant investment in this building alone, over $4.5 million, to bring together all of those activities in one place. This building is the innovation hub, and this is the center for all things innovation, entrepreneurship, and commercialization at UGA. Previously, we had all the activities disparate around the university, and we've brought them together in one place to help faculty and alumni move their projects forward. The Innovation Fellows Program is a new program which supports 2 faculty members per semester, where we actually customize the program for the needs of the individual faculty members.
Yeah, so I thought it was a great way of trying to see how we can get our technologies out to end users, and really explore the scope of commercialization and licensing opportunities for these technologies. Being in a lab, you're obviously very close to the science, but not necessarily as close to the end users and the application of the technologies. So I thought it was a great way to leverage some opportunities here at UGA to explore these avenues. Within CMaT, I primarily work with Testbed 2, which is developing these glioma on chip types of devices to look at how these tumors progress and how CAR T cells can target these tumors more efficiently. So applying it to the broader context of how it's going to help patients and clinicians is something that I think the Innovation Gateway as well as CMaT have kind of brought into focus for us.
We bring all of these different aspects together so that anyone who's got a new idea or a novel technology like those at CMaT, move them towards becoming a product which would be useful to people outside of the university and moving them into the public domain. So for instance, we'll help people work on how to assess what their technology could be useful for, and we then also give them the tools that help them move those projects forward so that they can actually become more relevant to the outside world, to corporates or to the public at large.
I'm very proud of the University of Georgia and the advances we've made at the level of CMaT. There's imaging technologies that is being married with metabolite biology in order to get a clearer and brighter picture of what's going on in the cell manufacturing area.